Alright, I understand that you guys have had the opportunity to read through these notes already. This is not meant to take the place of that, nor is this going to be simply an opportunity for me to read it out to you again. I do want you to listen as I go through this stuff with you right now, mainly because I want to make sure that you understand what we're talking about, and I'm going to kind of clarify a few things as we go through. So first of all, what I am going to ask that you do as we go through this is when you see brand new words, and there's going to be a few of them, I want you to highlight them so you can come back and get those defined in the lines next to the slides later on. Even if the definition is given on the slide, if it's a new word, for example, on this first one, endocytosis, exocytosis, those ones you should be writing down. Active transport, diffusion, osmosis, maybe you've heard them before, maybe you're like, I should write those down. And I would recommend actually diffusion and osmosis being two words that you definitely write down. Facilitated diffusion, if you understand what diffusion is, not a bad thing. All of these are, bottom ones here, are passive transport. The top three require energy. Active transport can be endocytosis or exocytosis. There's also various pumps that can be used. But the idea here is that all of the organelles in a cell and the cell itself are surrounded by this phospholipid bilayer, the cell membrane, and that's a barrier. And so to get the food and raw materials in, get the waste out, whether it's from organelles or from the cell itself, there's got to be some way to move in and move out. So diffusion is the first one. We chatted about this yesterday. The lovely smell or thought of the smell here. Yay. If we talk about here, when we talk concentration, number of molecules per unit area or volume, things that are high concentration, this is where we've got many molecules of the same type packed tightly together. This generally makes a hypertonic solution. This is one of those words you should be writing down. When we talk about low concentration, we're talking about a hypotonic solution. Now, I can't just say this one solution is hypotonic. It's not very concentrated, therefore it's hypotonic. I can't actually say that. When we talk about these two words, hypertonic and hypotonic, and then later on there'll be one more word, isotonic, it's always in comparison to some other solution. So if I have a solution of regular pop and watered down pop, the regular pop would be hypertonic, the watered down pop would be hypotonic. Or if I make some apple juice, have some apple juice, and I have one that is your normal apple juice, the other is apple juice that I poured onto ice because I want it to be cold, and then I let the ice melt, and now it's really watered down. The one that's low concentration, hypotonic. The one that is high concentration, hypertonic. But these two words are always kind of comparative terms. Right, so here's a happier version of diffusion. I open a bottle of perfume, eventually we all smell it. Put a drop of food coloring in water, it will spread out. It will always move from high concentration to low concentration. Highlight those words there. They're going to move down the concentration gradient until everything is equal concentration, or what we call isotonic. So theoretically, if I pour 2 glasses of apple juice from the same bottle of apple juice, both of them will have the same concentration. They would be isotonic. All right, so things that affect the rate of diffusion: temperature. Higher temperature means faster diffusion. Size, smaller molecules, faster diffusion. Concentration difference, the larger the concentration gradient, faster diffusion. Right? If it's 90% something and 10% on one side and 10% on the other, that's going to go a lot faster than if it's like 47% on one side and 53% on the other side. If we're talking gases, the higher the pressure, which means more packed molecules, means faster diffusion. Okay, cells, what does this have to do with cells? Well, think about the idea of blood flowing to the cells in your left big toe, or right big toe if you'd rather. That blood is high in oxygen, low in carbon dioxide, because it's coming from the lungs. Inside the cells, they are low in oxygen and high in CO2 because cellular respiration has been occurring. It's using up O2, it's making CO2. Oxygen diffuses down the concentration gradient from high concentration in the blood to low concentration in the cells until the concentrations are isotonic. Carbon dioxide really does the same thing. It goes down the concentration gradient from high concentration in the cells to low concentration in the blood until the two solutions are isotonic. And the blood continues to flow, which continues to bring more oxygen and take more carbon dioxide away. But that's about 20. Cell membrane itself is semi-permeable. That means it is picky about who gets in. It only allows certain materials to pass through. Anything that can squeeze between the phosphate and lipid components of the phospholipid bilayer can get through. Most things actually can't. Other examples of diffusion. You can get transdermal patches, and these would be medications that stick to the skin. They've got a semi-permeable membrane lining the inner surface of the patch, allows drugs to diffuse out of the patch at a slow, constant rate. So people who have heart disease sometimes will be given a nitroglycerin patch. Somebody who's trying to quit smoking may end up getting a Nicoderm patch. Somebody who has kidney failure may end up on dialysis. How does dialysis work? Well, in hemodialysis, a person's blood is pumped from their arm through a pump, and it goes through they call it a dialyzer. And it's going to be going eventually through a hemodialysis machine. And the dialysis tubing, which the blood flows through, is going to be immersed in a salt solution with a salt concentration similar to blood. Pores in the dialysis tubing allow small dissolved waste molecules to diffuse out of the blood. The large proteins in blood cells are forced to stay inside. What this does is it gets rid of any of the waste materials in the blood and allows the blood to become cleaner. Less waste means that they're able to continue living even though their kidneys aren't properly working. Okay, so that was diffusion. Osmosis. This is the movement, and this is actually just a special kind of diffusion. It is the diffusion of water through a semi-permeable membrane. It's passive transport. Water, just like solid, liquid, and gas solutes, is going to move until the concentration of solute on both sides of the membrane is the same, as in it's isotonic. But if water is the only thing that moves, let's try that again. If water is the only thing that can move across the membrane, that means it's going to look different and sometimes be a little bit confusing, because when solids or dissolved stuff, so solids or dissolved stuff move by diffusion, they move from the hypertonic, where there's more of them, to the hypotonic, where there's less of them. When water moves by osmosis, it moves from the hypotonic side. This is actually hypotonic, but that means that there's more water and less dissolved stuff. And water would move from the hypotonic, where there's more water, to the hypertonic side, where there's less water. So it is just a special kind of diffusion, but it is a diffusion of water. Are there examples of osmosis in nature? Well, yes. Not that a grocery store is necessarily nature, but this is one example of what's going on here. So water is hypotonic compared with the hypertonic cytoplasm of vegetable cells. If we were to spray vegetables with water, water moves from hypotonic to hypertonic. Vacuoles, central vacuoles especially in the plant swell. Full vacuoles mean crisp vegetables, meaning not floppy and soggy vegetables, meaning vegetables we want to eat. All right, what if you watered a plant with salt water? So if we watered a plant with salt water, water would be hypertonic for salt compared with the hypotonic cytoplasm of the plant cells of the roots. Water moves from where there's more water to less water. It means from the roots back to the soil, and the plant dies because there's no water for photosynthesis. And if there's no water entering the plant, it means no nutrients move from the soil to the top of the plants. Plant cells need a full water vacuole to not wilt. This is called turgor pressure. There's a word you should probably underline. We're going to talk more about that in the next section on plants, but turgor pressure is something that you need to know about. Paramecium. Interesting creatures. These can live in a hypotonic environment, which means they are always getting excess water into them by osmosis, and so they actually have to forcefully expel water. They've got a special organelle and set of structures called a contractile vacuole. Otherwise they would burst. So they got to get rid of the extra water inside of them. All right, reverse osmosis. You might have seen this like Dasani water or various other things where you're getting filtered water. Water is filtered really through an artificial membrane containing very fine pores that block large particles such as bacteria and salts from coming through. And it's just the opposite way, right? Normally, water would move from where there's more water, which would be like over here, to where there's less water, which would be over here. But in this case, we're putting pressure on it. We're doing work, or adding energy to go against osmosis. Facilitated diffusion. This is the third type of passive transport, as in no energy required. And so in this case here, if we want to get large water-soluble substances into a cell membrane, they need to go through channel proteins or carrier proteins. These are words we should be underlining and defining later. This is still passive transport. And no energy is required. It's still diffusion. We move from high concentration to low concentration of the solute. Takes us to active transport. So if you think about materials moving down the concentration, they require no energy. This is like floating with the current in a river. Sometimes cells need to move material from low concentration to high concentration. So, like swimming against the current. Requires energy and specialized membrane proteins. Energy to power active transport comes from sugar burned in the mitochondria. Cellular respiration. The released energy is stored as ATP. You should at least highlight this. This is something, and get what ATP means, adenosine triphosphate. That's a good thing to rewrite. ATP in turn releases energy when it is converted to ADP, or adenosine diphosphate, as chemical bonds in the molecule break. Active transport works by energizing the carrier proteins, and they act like a pump. And they change shape to capture a solute molecule. Changes shape again, releases the solute on the opposite side of the membrane. Active transport can then be used to pump material into or out of cells. Your nerve cells have sodium-potassium pumps, which keep pumping sodium and potassium against the concentration gradient. Your kidneys move salt from urine-carrying tubes back into the blood to maintain your own blood salt levels. All right, and this takes us to then our last bits here with endocytosis and exocytosis. Some materials are too large to diffuse through the cell membrane or move through channel or carrier proteins, even through pumps. Some of them just can't fit. And so endocytosis is a way of the cell absorbing bigger material. And the cell membrane itself actually folds around the object and then into the cell. And the phospholipid bilayer or membrane forms around whatever the substance is, and it actually just forms a small vesicle or vacuole around it as it infolds around it. So this is how amoebas feed white blood cells. This is how they absorb HIV or other viruses as well, can be absorbed into the body this way. Exocytosis is sort of the opposite. The cell is going to expel material as the membrane of a vacuole or Golgi body fuses with the cell membrane. The contents of the storage sac or waste, which generally are cell waste, are dumped outside of the cells. Can also be neurotoxins, or in the human body, when we make neurotransmitters that allow nerves in the brain or in the spine or anywhere else but to talk to each other. The one nerve will make neurotransmitters, store them in vesicles. When it's ready to release them, those vesicles fuse with the outer cell membrane and will actually open up and release whatever was stored inside of them. But if E. coli makes neurotoxins, they would then be released by exocytosis and taken into healthy nerve cells by endocytosis. This is obviously a problem. You don't want to get E. coli. It's nasty. You should understand what this word means. You should highlight that, and you can find a definition later on. And that takes us to the end here. I thought I had one more, but this was the last one. Check Google Classroom. There will be an assignment for you, but before you get to that assignment, you need to get all of these definitions down. And if there was other words that you're like, I'm not sure if I'm sure about that word, get them defined as well. You can use your textbook. You can use Google. I would recommend you actually write them down, though. Don't just copy and paste them into a Word document. The process of you writing them will help you to remember them more than just copying and pasting. All right, thank you.